This is the video for DNA Structure and Modeling Project by Jaina Lee, Laura Nolasco, Ashley Lee, and voiceover by Vincent Cooper. Enjoy.
DNA is a double helix structure with two anti-parallel strands that are made up of nucleotides. Nucleotides consist of deoxyribose sugar, phosphate groups, and nitrogenous base. In our model, the sugar and phosphate group will be represented by Twizzlers, serving as the backbone of the DNA connected by covalent bonds. Meanwhile, the nitrogenous bases will be represented by 4 different colors of gummy bears. Red ones will represent adenine, dark yellow will represent thymine, orange will represent guanine, and light yellow will represent cytosine. The beginning point of DNA replication is called the origin. The enzyme helicase breaks down the weak hydrogen bonds between the bases and separates the two strands, forming a replication fork. SSB protein, short for single-stranded binding proteins, will attach to the two individual strands to keep them from rewinding. And near the replication fork, the enzyme topoisomerase presents supercoiling. After that's done, primates synthesize primers made of RNA molecules that are complementary to the strand. This provides DNA polymerase III a point to start making the new DNA strands. DNA polymerase III works in a single direction from 5' to 3'. Thus, it adds onto the 3' end to make the bulk of the new DNA. Because of that, DNA synthesis of one strand will go in the same direction as the replication fork, called the leading strand. The other strand will go against the direction, causing it to lag behind, which is why it's called the lagging strand. Primates needs to make multiple primers on the lagging strand because it is synthesized in multiple short fragments called the Okazaki fragments. The gap between these fragments will later be sealed up by another enzyme called ligase. DNA polymerase I plays the role of proofreading the newly synthesized strands, as well as removing the RNA primers and replacing them with DNA. The new strands are complementary to the old strand and run in opposite directions. That is our model and description of DNA replication.